Hello and good evening, everyone present. I think we can start now. If more people join, I will admit them accordingly. Just let me know if you can see the presentation or not. The presentation is visible. You can type
Yes,
in. Yeah. So, good evening, everyone. Welcome to week one of the PMRF-NPTEL lecture or Animal Physiology. In these sessions, what we'll be doing is that we'll be covering various assignment questions that were dealt with in the previous years and go through them and try to understand the concept behind the questions and better emulate whatever was given as the weekly lectures in the main course. So we will be mostly solving various assignment questions that were previously done in the previous years. We will not be going through the assignment questions of this year. So this is week one. I will just quickly introduce myself. I am Anubhav Chatterjee. I am a Prime Minister's Research Fellow and PhD scholar at Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering at IIT Kanpur. So I believe that I am audible, right? Yes, I am audible, correct? You can type in the chat if you want to. Okay. So the first week of the lectures concluded with on various topics. The takeaway from the week's lectures was that there are various levels of organization that we have already seen: the molecular level, the cellular level, and the tissue level. And we are trying to understand physiology at its most basic, how exactly life has its different functions and how exactly different organs and organ systems work. And that is what we are going to focus on this week. So if anyone has any questions from the lectures, they can actually we can address that beforehand before going into the assignment questions. If anyone wants to know anything about the topics dealt in the lecture, if they had any doubts.
sir, there is no specific doubt yet, but I am having question about can I get the notes for the first week as well as the coming weeks?
The notes, the presentations that are being talked, like we are discussing. So these presentations will be uploaded to a folder that will be available to you by the end of the week, I guess. But you will probably be getting from me because I am doing it on Tuesday. So today we're going to discuss week one only, and you'll be getting the PPT for the week one by the end of the week. Similarly, for week two, you will get it by the end of the next week.
Sure, sir.
Okay.
And is
Yeah.
the date for examination?
The date for examination, I think, is 20. It is somewhere at the end of August or in the starting of October. I will have to check and let you know.
Even in the portal 26th of October, right?
Yes.
But before you go to just register yourself for examination portal, no?
Yeah.
So above, on the top of the above, they are written 26 December form. Like, so whether I am just filling the form for 26th of December form or for 26th of October, there is a confusion between that if I am filling the form.
Okay. Let me check once.
So, December form on the top.
Let me check once.
And sir, for the animal physiology, we are getting the domain certificate.
A domain certificate.
Yeah.
I don't think so, but I will talk with the people at IIT Madras and let you know if you are getting a domain certificate. But I think you will only get an exam certificate at the end after clearing the course.
So, will the certificate work
For what exactly?
like to uh, for my profile
Yes, it will work. It will definitely work
or
for your profile.
domain certificate is only important?
Yes. What is that?
See, basically, some people are getting domain certificate and some are getting the basic certificate, which is provided by the SWAYAM app.
Hmm.
Because we have done the what is it, particular course. So, what is the weightage of that domain certificate and what is the weightage of our certificate?
Okay, I don't think I know. I do not have much idea on this. You can actually try and ask this in the forum that is present on the PMR on the NPTEL course site. I think they will be better able to help you. The guys that are running the course itself, they have a better idea. They will have a better idea of how exactly this is working. Or as far as I know, you can actually get exam certificate at the end. But about the domain certificate, I do not have much idea. What I can do is that I can mail once and then let you know in the next class what exactly is going on. You can what you can do is that you
So,
can write down whatever you are trying to ask in the chat, and I will ask that in the mail.
but is it totally important to have a domain certificate?
I don't think so. The domain certificate is that important because previous courses that I have done in NPTEL, they always give out exam certificates at the end of the course, not domain certificates.
Okay, sure.
But you can write down whatever your questions are in the chat, and that I can ask via mail to the PMRF the NPTEL coordinators, and get back to you on that.
Okay. Today I had mailed about the notes. Okay. So now you had cleared my doubt about notes. Okay.
Yeah,
So
yeah.
that notes will be uploaded till the end of the week.
Yes.
So before this only I had mailed to NPTEL group.
Okay.
So might be they will receive my mail or something.
Okay. So they might answer you directly to your saying. Yes, that's also correct. Yeah, you
So,
can do that. Okay. Anything else regarding the coursework itself? Anyone wanted to ask anything about that? Hello. Anyone wants to ask anything about the coursework? If not, then we can start off uh, with the assignments directly. So how we are going to do this is that we are going to look at one of the assignment questions and we are going to first try and answer this. So this is a MCQ ABCD answer type of question. You can just try and answer this and put whatever you think is the correct answer in the chat. We will then have a discussion about the question and the concepts involved, and then we will answer the question. So the first question that we have today is that dash refers to the relatively stable state inside the body of an animal. The options are homeostasis, equilibrium, fluid balance, and organ symmetry. So ABCD, there are four options. Whatever you think is the right answer to this question, you can put it into the chat as a ABCD option. So Shayalu says A, homeostasis. Priya is also A. Akansha is A. Vinayak says it is B, equilibrium. Bisu says it's A. Aruna says something else, which I So Aruna, about this, I think once you pay the amount, it is for that particular cycle of exam only. It will not roll over to the next one. So unfortunately, I think you will have to take the course again if you want the exam certificate at the end of it. But again, you can actually mail directly the NPTEL in an NPTEL group, or I can mail for you and ask if it is a problem or not.
So do we have offline exam or online exam? Or
For NPTEL, it is a it is always a offline exam. You will have to go to a exam center and you will have to do a like MCQ question paper there as well. This will be very similar to what the assignments are. So that is why assignments are very important as well. So you will get prepared for whatever the final exam will be like. And the assignments also carry a lot of significant amount of the grade, and you will have to have like a basic amount of marks in order to pass the final exam. That is, I think, clarified in the main site website. You will have to check, but I think it is you will have to score more than I think it is 25 on the assignment and remaining on the final exam to have a overall score of 40 or above to get a certificate. Aruna says A, Pitambika says A, Varishtha says A, Ramana says C. Aruna is asking when that exam will be. This you will have to log in again and like you have to register for the new course and then check when the date of that exam will be. It will be present on the site. It will be present in the body of the website itself when that exam is happening. Is that fine, Aruna? But I think most of the NPTEL exams occur at the same time at the end of October, so it will happen at that time only. Okay, so we have gotten a lot of answers as A and some answers as B and C. Anyone else wants to attempt the question, they can quickly put into the chat what do you think is the correct answer. Anamika says it is A, homeostasis. Okay,
sir, there is another question
yeah,
regarding the exam.
yes, go ahead.
We are in particular center, okay.
Yeah,
So we will within the state or out of state.
so when you fill the exam form, they will give you options for which centers that you require that will be easier for you. I think there are 3 to 4 options, and you will be given your preference within those options. If your primary center that you have given that you want to go is not available, then you will be put down to the secondary center. But you can choose which centers are available. Depending on where you stay, it might be in your city, but also depending on where you stay, it might be out of state as well.
Sure, because for exam we should like go from one state to another to
Yeah, that is difficult, I know. So, what used to happen is that I have known people who have come across states to give exams at the same center because they did not have a center in their state. So that's difficult, I know. But you will have to check that. You will have the option to check that and see if you can appear in the exam at the correct time or not, and then proceed accordingly.
okay, sir.
So Aruna is asking what are the use of the certificates. Majorly, the use is that you can put that into your CV, and that will boost your CV that you have actually done an official course via NPTEL. If you do not have a certificate, then you can still say that you have done the course, but you will not have a certificate to show for it. So in official areas, that is a bit iffy when they require you to submit certificates for whatever is mentioned in your CV. So grabbing an NPTEL certificate is very important if you are putting a lot of effort into attending the courses, doing the assignments, and preparing for the exam, because that will actually help you in your CV and going forward as well. Okay, I think a lot of people have answered. So we'll go ahead and answer the question. So first, I'm going to have a discussion on what exactly is the relatively stable state inside the body of an animal. So the first thing you have to understand is that our body is very strictly regulated in various aspects. The temperature of the body, the pH balance in the body, the salt concentrations inside our body, the hormone levels, the sugar content in our blood, everything is very tightly regulated. The reason is that all of our cells are optimized to work efficiently at an optimum temperature and various other pH conditions. And if you go outside this range, then you will have problems. So our body tries its best to always remain in this stable state. This stable state is known as homeostasis. So homeostasis can be divided into two words: homeo meaning unchanging, and status meaning stasis meaning state. So unchanging state in which our body always wants to remain in itself. So homeostasis is a property of the human biological system where self-regulating processes tend to maintain the balance for survival. The regulation takes place in a defined internal environment. So always your body is trying to survive the best it can in according to the stimulus it receives from outside. So if it is too cold outside, you start shivering to increase your body temperature. If you are eating something, then your blood sugar rises, but it should not rise too much. So you have different enzymes that lower the blood glucose. If all of these go haywire, then you have problems such as hyperthermia, hypothermia, or diabetes, as we know. So how exactly is homeostasis, or the unchanging state the body wants to live in, maintained? This is maintained by something known as a feedback loop or a feedback mechanism. What happens is that our body has various receptors or sensors that are present throughout our body. These sense the temperature, the touch sensation, the blood pressure, the pH, various things. And once, if there is some change in the external environment or the internal environment that causes either too much or too little of something to occur, that changes the homeostatic balance, that produces a stimulus. The stimulus is an external change that is occurring in the system. This is detected by a receptor. This receptor sensor then provides this information to a control center. This control center is often our brain, could be parts of the central nervous system, could be parts of our glands which control hormones. This control center then integrates this information and then decides what is the best way to go forward to re-establish homeostasis. So it integrates the information and gives the command to an effector. This effector is could be the nervous system itself, the various glands that are present in our body that release hormones, or various parts. So these are the major two parts. Either it will be a hormonal or a nervous system regulated effect, which finally the response of the effector feeds back to the influence of the magnitude and returns to variable homeostasis. So once the effector has done its work, you should return to homeostasis. Depending on how much the initial imbalance was, the effector will try to double its effort in order to bring it back to its to the homeostatic state that much. For example, say it is cold, like around 15 degrees Celsius. So in that situation, your body is not very worried about anything about the cold from outside, but it still needs to raise its temperature to maintain its normal homeostatic temperature of 37 degrees Celsius. So body starts to produce heat by thermogeneration, which is mostly done in the core of the body. But once temperature starts decreasing even more, say 10 degrees or 5 degrees or all the way to zero, then your body starts something known as shivering, in which the muscle starts to vibrate in order to produce heat. So as much as the imbalance, the effect will be that much. So in our body, various organs or various kinds of homeostatic balances are maintained by different parts of the body. So the hypothalamus is the thermostat of the body that maintains the temperature and the osmotic pressure. The kidneys maintain the water balance, pH and salt concentration. Blood throughout the body helps in heat distribution. Skeletal muscles, via shivering, also help in heat generation and distribution. Pancreas is an organ in our body that produces which? Enzyme? Pancreas produces which enzyme? Does anyone know? Anyone has any idea?
Pancreatic enzyme.
Pancreatic enzymes. Yes, pancreas produces the pancreatic enzymes that help in digestion. But there are two very interesting enzymes that we hear a lot about, which are present in the islets of Langerhans, if anyone remembers.
Insulin.
Yes, insulin and glucagon are two enzymes which maintain and help regulate the blood sugar level in our body. These are sensed and produced by the pancreas. And finally, evaporation of the water from the sweat glands helps in regulate body temperature. This is the reverse process that we are discussing. In case the temperature is too much, we start sweating, so that evaporation occurs on our body surface and we can decrease the temperature of our body. So, yeah, insulin, glucagon. Ramanna and Aruna, yes, you are correct. These are insulin and glucagon. Okay. So, for example, one of the feedback mechanisms are shown here. When we eat, then our blood glucose rises. But in blood glucose, what happens once the blood glucose rises is that all of our cells start taking up sugar at once because cells love glucose. The moment they start taking up a lot of glucose, that is a problem for our body. So what happens is that pancreas releases insulin. This insulin helps the liver take up glucose and store it. It stores it in the form of glycogen, which is a bigger molecule which cannot be utilized by the cells. And once blood glucose, this helps the blood glucose to be decreased because liver is taking up all the glucose inside it, and then you have optimal glucose and homeostasis maintained in the body. Is this fine? Anyone has any questions about homeostasis? Because this will be an important concept moving forward throughout this class as well as throughout our course, because homeostasis is one of the most important concepts around which physiology is based. So you can take a minute and think about it if you have any questions regarding this.
Or what is the difference between animal physiology and human physiology?
So when we are discussing about animal physiology, we are actually defining, we can discuss about the entire animal kingdom. So we will be discussing about fishes, we can discuss about various model organisms such as mice or frog or fish even, such as zebrafish. So we will be trying to study. So most of the time what we do is that when we look at a difficult subject, we try to study it in its simplest forms first and then make our understanding and develop from there to higher organisms. Human physiology will be for something that we are aiming to study. Okay. Human physiology is something that, give me a second. Okay. So human physiology is something that we are aiming to study in this entire course, but we'll be starting with basic concepts and build up from there. We'll start with simpler organisms, understand how physiology works in them, and then go forward. Ramanna is asking if I know Tamil. I do not. Sorry, Ramanna. You can ask it in English if you want. I know Hindi, if that helps.
चलेगा सर.
चलेगा ना? ओके. तो कुछ भी पूछना है तो ये हिंदी में पूछ सकते हैं एंड हम उसके लिए जवाब दे देंगे आराम से. ठीक है.
From Mumbai. So
You're from
fine.
Mumbai. Okay, okay, that works. Yes. Okay. So anything else that anyone wants to ask about homeostasis, you can ask it directly or put it into the chat. Okay, I don't think so. So the answer to this question is homeostasis. Homeostasis refers to the relatively stable state inside the body of an animal. So for the other answers, equilibrium is something that is similar, but equilibrium is a much simpler state of affairs. Equilibrium is when we are talking about a very simple system in which there's input, there's output, and you try to make a balance between it. This is very easily understood in case of chemical reactions. If you have heard of the La Chatelier's principle, equilibrium is maintained in these conditions. But homeostasis is much a broader term that is more suited to physiology. Fluid balance and organ symmetry are not really close to the answers we are looking for. So the first question's answer is homeostasis. Anything anyone wants to ask about this?
Hi, sir.
Hello?
Yeah, am I audible?
Yes, you are audible. You can go ahead.
Sir, in the last slide, you have shown that about the example of blood glucose increasing while having the food.
Yes, yes, yes.
So I was not getting the right connection over here, so I missed some points. So is this a positive feedback loop or a negative feedback loop?
Okay, that's a very nice question. So we will be discussing that in detail in one of the upcoming questions, but I will just give you start like a simple answer. When see there is a stimulus that is happening, so the external environment is pushing the homeostasis to one end. So for in this situation, the blood glucose is rising. That is the stimulus. If our overall feedback is in the direction of the stimulus, then it is a positive feedback. If it is in the opposite direction of the stimulus, then it is a negative feedback. So first blood glucose rose, that was the stimulus, and the end is where blood glucose is decreasing, so in opposite directions. So this is a negative feedback. Is that
Okay,
fine?
sir.
Okay. So we'll be going forward with the next question. This is a fun question. The Arctic fox, which is a cute little white colored fox, uses what as extra insulation when it curls up to sleep in cold weather? The options are its fluffy tail, its ears, its nose, or its mouth. Before we answer this, I will just show you a picture of this fluffy animal. So this is our Arctic fox. So ABCD, you can answer the question. This is very
A. Fluffy tail.
A. Fluffy tail is one of the answers we got. We have a lot of A's. Okay, so you are correct. It is the fluffy tail of the Arctic fox. It is what you can see right here. It covers itself up in with this fluffy tail. So this is also an example of maintaining homeostasis. Other animals. So this is a good point. Primates are can sweat through their skins. Many animals cannot, which actually gave us a evolutionary advantage to become the prime predators around the world. Other animals develop different kinds of organ systems to fight cold or temperature. For example, Arctic fox uses it for a fluffy tail. Other animals have fur. Some animals, such as the bear, has a thick layer of insulation or fat around its body. Seals also do that to maintain combat a very low temperature. So different animals have different ways of dealing with different external stimulus, but all of them want to maintain homeostasis. For example, elephants also have a very cool trick which they use to maintain homeostasis and reduce the temperature of their bodies. Does anyone have any idea what exactly elephants do? Does anyone have any idea what exactly elephants use to decrease their body temperature? They use their trunk. Ears is the correct answer. So elephants, if you have seen them, they have huge ears that are very that have blood vessels going all through them. So what they do is that they increase the blood flow in the blood vessels in the ears, and that blood flow and the vessels are dilated. So a lot of blood is passing through the ears, and they use that huge surface area to radiate heat outside of their body and decrease their temperature. So for this question, it was very easy. Artifact for the Arctic fox uses its fluffy tail as extra insulation when it curls up to sleep in the cold winter. Okay. Another question on the temperature of the body. This time we are talking about the human body. Approximate short-term non-lethal limit for body temperature in Fahrenheit is the following options. It is 1 to 5, 10 to 50, 65 to 110, or 115 to 200. What is the approximate temperature that our human body can survive in Fahrenheit? So there are four options A, B, C, D. You can put in whatever answer you think is correct. Vashishta says it is C, Aruna says C, Priya says C, Akansha C, Shannon C, Bisu C. So we have a lot of C's. Amanda is also C. Anyone else wants to go have a try at this? Okay. Okay. So I guess we'll go ahead and talk about this. So maintenance of the body temperature is done by the organ known as hypothalamus, which is present in the middle of our brain, and it's a very small organ, but it acts as the thermostat of our entire body. So how exactly thermoregulation works? There are two parts of it. How exactly the heat gets inside our body, and how exactly it is radiated outside. The first part of it is that we receive most of the heat around us via infrared radiation and convection. So the temperature, the air around our body is heated up, and that causes heat to be transferred to our body. How exactly do we reduce the temperature in our body? There are two ways. First is known as vasodilation. Vasodilation is the increase in diameter of blood vessels, and this occurs majorly in the top layers of our body. So our skin and every organ on the top layer starts increasing blood flow inside. What this does is that, so if you have an idea about the physics of it, then a bigger surface area allows for higher radiation of heat. So once there is a huge bigger surface area which is produced by vasodilation of the blood vessels, then more heat is radiated outside of our body. So we decrease the temperature of our body. The second way it is done is that we start sweating. So sweating, uh, what it does is that it causes evaporation to occur on the surface of our body. Evaporation always decreases the temperature of the surroundings. So our body actually puts heat out of it using this particular trick. The opposite also occurs, in which we try to maintain the temperature of our body in cold environments. This is done via vasoconstriction, the opposite of the previous one, in which the blood vessel starts constricting, lowering the surface area, preventing radiation outside of heat outside of our body. The second thing it does is that it stops sweating completely and starts shivering. Shivering is the process in which the muscles of our skeletal muscles of our body start vibrating in order to produce heat via friction. And all of this is controlled by the hypothalamus, which is known as the thermostat of our body. So here we have an example of the various temperatures in which the human body can survive. The normal temperature is from 100 to 96.8 degrees Fahrenheit, or around 37 degrees Celsius. If we start increasing the temperature, we reach pyrexia, or what we know as fever, or hyperpyrexia, which is a very dangerous condition in which cell death starts to occur. The opposite is known as hypothermia. We can survive hypothermic temperatures much better, where our body temperature decreases, but severe hypothermia can also cause a cardiac and respiratory functions, our cell death, and so reduction of blood flow towards the extremities. So in very severe conditions, what occurs is that our body goes into survival mode. It only because the blood is carrying heat to the surface of our body in extreme cold conditions, the blood starts to only circulate in the core of our body, where there is higher chance of the heat being contained in our body, and this causes heat blood flow to stop in the extremities due to extreme vasoconstriction. This produces an effect known as frostbite, but this occurs in very low conditions. Okay, Aruna asks, temperature increasing or decreasing in our body is a negative feedback. Yes, the temperature increase or decrease is a negative feedback, because once, if you are in a cold condition, body starts shivering in order to produce heat and raise the temperature. So first, the initial stimulus was a lowering of temperature. The final effect is increase of temperature. So you see these are in the opposite directions. So it is a negative feedback loop again. So air temperature control in our body is a negative feedback. Okay. Anyone has any questions regarding this?
No, sir.
Okay. So the approximate short-term non-lethal limit is from 65 to 110. Everyone was correct when they suggested this. It is option C. In Fahrenheit, it is 65 to 110. Okay. So this we have already discussed. Any homeostatic process that changes the direction of stimulus, that it is opposite to the direction of stimulus, is a positive feedback loop, negative feedback loop, equivalent loop, or a distributive loop. You can put the answer in the chat, A, B, C, D, whichever you think is the correct one. We have B, B, B from Aruna, Kanksha, and Bisu. Anyone else wants to try? Vinayak also says V, B. Vasu also says B. Anyone else? No, this is okay. Avantika also says B. So we have discussed this for some time now, that in case of a feedback loop which maintains homeostasis, if the variable changes that is occurring is towards one direction and the effect is in the opposite direction, then it is a negative feedback loop. If the change is in one direction and the effect is also in the same direction, then it is a positive feedback loop. So here we have an example of both, in which if there is a too much imbalance, then we have an effect of bringing it back to the variable homeostasis, and too little also does the same. One of the important factors is that negative feedback loops is something that we will mostly see in all of our discussion. The reason being is that positive feedback loops can produce a vicious cycle of continuously increasing stimulus and effect. For example, if say temperature control was a positive feedback loop, then increase in temperature of our body will cause receptor to sense it, control center to integrate it, and effector to keep on increasing the temperature, which still contributes to the stimulus. So you have a continuously increasing feedback loop, which is the positive feedback loop. This causes a vicious cycle to occur, which can overall contribute to change in homeostasis, or coming away from homeostasis. Someone raised a hand, or did someone have any questions from this? Okay, I don't think so. There are any questions. So anyone wants to ask anything about the feedback loops again? Then we can discuss it. Okay. So here we'll be discussing about a bit about the positive and the negative feedback. So one of the examples that we had seen previously about the glucose imbalance, we'll be discussing that again. So the normal blood glucose level is around 90 mg per 100 ml in our body. But after eating something, the first thing that happens is that blood glucose rises spontaneously, and this rise is sensed by the pancreas. After pancreas senses this rise in glucose, it produces insulin. The insulin causes various cells in our body, which are the tissue cells, to start the uptake of glucose, as well as the liver to take up glucose as well. Both of these processes take the glucose out of the bloodstream and store them inside the tissues or the liver. Liver stores it in the form of the insoluble carbohydrate glycogen, which overall what it causes is that reduction of blood glucose, and blood glucose falls to the normal level. So there is a rise in blood glucose, and final effect is a decrease in blood glucose to the normal levels. So this is an example for negative feedback. Another example is what happens when the blood glucose decreases. So when you have not had food for a while, then the blood glucose will start decreasing, because there is only so much glucose present in the blood at that time, and cells are continuously using up glucose in order to function. So once there is decrease in blood glucose, this is also sensed by the pancreas. Pancreas then releases the alternate enzyme, alternate hormone, glucagon. What glucagon does is that it stimulates breakdown of glycogen stored in the liver into glucose, and then subsequent release of glucose from the liver into the bloodstream. This again increases the glucose and brings back the normal homeostatic blood glucose levels. But again, if you see right here, the initial stimulus was decrease in blood glucose, and the final effect is increase in blood glucose. So this is again a negative feedback loop. Is this understood? Anyone has any questions from this?
No,
Anyone?
sir.
Okay. If there are no questions, so this is a negative feedback loop. Now let us discuss a positive feedback mechanism. So positive feedback, one of the examples includes blood clotting. So say there was a tear or a break in one of our blood vessels, and blood started pouring out, which can occur via a scrape on your skin or any injury that you receive. The stimulus is then the break or tear of blood vessel of the wall. What this does is that the cells which are damaged, as well as the cells surrounding the break, they start releasing various chemicals. These chemicals do two things. First, they cause platelets, which are small fragments of cells present in our blood, to start to adhere to this break. So everything around the breaks, the platelets start in sticking to the walls of the blood vessels, and they also cause the platelets to work up and release various chemicals. These chemicals start forming a mesh-like structure, which is like a fishing net all along this blood vessel. Plus, there is a positive feedback loop which causes these cells and platelets to release even more chemicals. So even more platelets start surrounding this break, and the final fishnet structure becomes even more dense, and we have a complete blockage being created by this mesh, as well as the platelets and cells stuck in it. So increase in the first release of the chemicals, start release of more chemicals, start release of more chemicals, more adherence of platelets and cells to the broken vessel wall, and finally what this causes is that formation of a clot, which is a structure which is mostly the platelets as well as the mesh-like structure that they have formed, and these clot starts to squeeze and bring the vessel walls closer to each other to close the break in the blood vessel and seal off the tear. This is how blood clotting works. So in this case, the initial stimulus is the break and release of chemicals which are known as coagulants, which help in blood clotting, and these blood clotting agents release more and more blood clotting agents, which actually help in progressively more complex mesh and a complete blockage of the tear to occur. In this case, this is a positive feedback mechanism. Is this fine? So clotting of blood is a pretty complicated process that we will probably be dealing with towards the end of the entire course. But this overall, this is a simplistic representation what exactly happens. Any questions from this?
No, sir.
Okay. So any homeostatic process that changes the direction of the stimulus is a negative feedback loop, as everyone has answered beforehand. Okay, so moving on, the next part of the next question is, which of the following parts of the brain is majorly involved in temperature regulation? The options are the visual cortex, hypothalamus, nephron, or the olfactory bulb. So it is ABCD option. You can put in your answer, whichever you want think is correct. Aruna has said it is B. Anyone else? So we have a lot of B from Aruna, Sharan, Bisu, Ramanna, Akansha, and Sanjana. I think someone raised their hand. Does anyone have a question that they want to ask? Any questions from this? I don't. So people are pouring in the B. Sanjana, Madhumita, and Ashna has also said B. Anyone else that wants to ask anything or put in an answer? Maheshwari also says it is B. Anyone else wants to answer this? They can. Okay, for some people who have just joined, so we are discussing these questions from previous year's assignment of the first week of Animal Physiology course. The question is, which of the following parts of the brain is majorly involved in temperature regulation? There are four options: visual cortex, hypothalamus, nephron, and olfactory bulb. So if you have an idea what this could be, then you can put it into the chat as everyone else has. Vinayak also says it is B. We'll wait a minute for any other answer, then go forward. Okay. So we have been discussing for a while now that the temperature set point or the temperature thermostat in our body is this structure which is known as the hypothalamus, which is present at the base of our brain. This structure has various sensors which sense the temperature of our body as well as some other things such as the osmotic pressure of our blood. But here we are going to discuss about the normal body temperature maintenance by the hypothalamus. So all of our bodies, the hypothalamus present in our body has something known as a hypothalamic set point. This hypothalamic set point determines what the temperature the body will always try to maintain. So in normal condition, this is at 37 degrees Celsius. When the hypothalamic set point, the body temperature rises above this hypothalamic set point, then there are multiple problems. There are multiple effects that occur throughout the body to reduce the temperature back. The first thing that occurs is that brain signals dermal blood vessels to dilate and sweat glands to secrete. Dilation of dermal blood vessels increases the surface area of the blood vessels and help in of radiation of heat outside of our body. And sweat glands that secrete sweat help in evaporation and again decrease the temperature of our body. Body heat is lost to the surroundings and body temperature drops towards normal. In case of the opposite condition in which the body temperature drops below normal, then brain again sends signal to the blood vessels and the sweat glands, but in the opposite direction. In this case, the dermal vessels constrict, preventing like reducing the surface area for radiation to occur, and sweat glands remain inactive. And if this is enough, then to maintain body temperature, then body temperature rises and everything is fine. If it does not have enough of an effect, then the nervous system comes into play and starts the muscles to contract involuntarily and start to vibrate, which we perceive as shivering, where the muscles starts to cause friction amongst themselves to produce heat. This muscle activity generates body heat, which again increases the body temperature, causing it to come back to normal. So this is how body temperature regulation occurs in us. Any questions about this? Okay, so this is what happens in normal condition. Can anyone? Aruna is asking, after completion of the online class, can we get notes or text back? Yes, Aruna, as I said, I will be uploading it to a folder that will have all the entire PPT for you to access, as well as the recording of this session that we are conducting will also be available on YouTube. So you will have the channel link for YouTube as well as the PPT, and you can go through both of them and then come back, or like, and go through both of them and then understand the lecture better at the next point. And you can come back and ask anything you wanted to ask, right in the NPTEL forum or in the next session.
Yes, sir.
Okay. So now what I was asking is that this is what happens when we have a normal condition. But does anyone know what happens if there is one particular condition in which our body by itself raises its temperature? What is that condition? The condition in which our body by itself raises its temperature. It does not stay at 37 degrees Celsius or 98 degrees Fahrenheit. Ramanna is correct, it is fever. In case of fever, our body raises its temperature. And why does that happen? It happens because, as we have said, that our cells and tissues in our body work best at the optimal temperature of 37 degrees Celsius. But that is also the case in case of pathological cells, such as bacteria or various parasites that are infecting us. And in that condition, our body raises its temperature to kill off these bacteria because they also cannot survive at a higher temperature. In that situation, what happens is that our hypothalamic set point in our hypothalamus is raised. So this hypothalamic set point changes to 100 degree or 104 degree Fahrenheit, which then causes the body to try and achieve that temperature via shivering or just raising the core temperature of our body. This happens in case of fever. So our hypothalamic set point changes. That is why during fever our body does not automatically try and maintain our temperature and lower it.
Aruna Muruga.
Hello, Vinayak. Uh-huh. Thank you for muting yourself. So anyone has any questions on this?
Okay, sir.
Okay. Anyone else has any questions to ask from here?
Okay.
Okay, so the answer to the question, which are the following parts of the brain is majorly involved in temperature regulation, the answer is hypothalamus. Okay, now we come to the next part, next question. Dash is the process of maintaining salt and water balance across membranes within the body. And
Osmoregulation.
yes, you can put it into the chat as well. So the options are distillation, osmoregulation, salt acid balance, and pH. So we have a lot of answers coming in. Bisu, Akansha, Aruna, Varshishta, Sanjana, Madhumatha, Ramanna, Muskan, Ashna, and Priyas. All of them say it is B, osmoregulation. Anyone else wants to try? Sharon also says it is osmoregulation. B. Vinayak also has said it is B. Anyone else? Sareshwati also says it is B, osmoregulation. Okay. Anyone else wants to try an answer? We have a lot of people in the meeting. We can have a lot more answers. Anyone else wants to try and answer this? Okay. So we'll start discussing about osmoregulation. So the answer, everyone was obviously correct. The answer is osmoregulation. It is the phenomenon by which our body maintains a particular salt and water balance across its various tissues and cells. But in order to understand osmoregulation, we have to understand a concept known as tonicity. So all of our body cells and normal cells that survive out in the wild as well are present in something known as a isotonic solution. Isotonic solution is one in which the concentration of salts present inside the cell is equal to the concentration of salts outside the cell. So this concentration equivalence between the intracellular side and the extracellular side causes no movement of salts or water through the cell, and this will cause the cell to remain in its natural condition. So in an isotonic solution in which the concentration is similar, the cells are happy and stay in the shape they want to stay in. But in case of there being a disbalance of concentration of solutes present inside the cell and outside the cell, there occurs a process known as osmosis. Osmosis occurs, what happens is that water always tries to travel from a less concentrated part to a more concentrated part. If the cell is in a hypotonic solution, in which the water level, the concentration of solutes outside the cell is less than the concentration of solutes inside the cell, water starts to get in the cells from all through the membrane, and this causes the cell to swell up. This swelling up can be dangerous for the cell. If it continues, it can rupture the cell because of too much pressure. This occurs in a hypotonic solution in which the concentration outside is less than the concentration of solutes inside. The opposite condition can also occur, which is known as a hypertonic condition, in which the concentration outside of the cells is higher compared to the concentration of solutes inside the cells. What this causes is that because there is less concentration inside, water tries to go out of the cell. Because most of the volume of the cell is made out of water, because the cytoplasm, the internal environment of the cell is aqueous, so it is mostly water. Once it goes out of the cell, then the cell starts to shrink in size. This collapse is also very dangerous because it can also kill the cell. So we either have a hypertonic solution, isotonic or hypotonic. The cells are happy in an isotonic solution. This is mostly in case of, before going forward, is everyone okay with this part of it, tonicity, what it is? Everyone is fine with this? Okay, Sharon says yes. Okay. So most of the animal cells love to stay in an isotonic solution. The one exception to this is plant cells. Plant cells have an extra layer. If you would know, the plant cells have an extra layer outside the cell membrane known as the plant cell wall. What a plant cell wall does is that it produces solid architecture. So, in case of a hypertonic environment in which plant cells mostly stay in, the cell membrane bulges out, but it cannot bulge out too much because of the uh, plant cell wall, which essentially confines it into a stable structure. In this condition, where the cell is trying to expand or swell but is prevented from doing so because of the plant cell wall, is known as turgidity. This is something that plant cells do and they are very happy with it. But animal cells cannot do that and they always love to stay in an isotonic solution. Now the next question comes is that who exactly maintains the tonicity or the salt and water concentration of our blood, which is the most abundant extracellular fluid that is going around? The answer is it is done by the kidneys. Kidneys are the two guys that help us in absorbing water as well as maintaining the salt concentration outside the cells, out the salt concentration in the extracellular blood, which may make sure that the cells of our body stay in isotonic condition and are very happy staying in it. So we are going to discuss about this. So in an initial condition, we believe we are starting with the condition that the water content of our blood is normal. If you drink too much water, then the water content of our blood rises. So the extracellular part becomes less concentrated. So this is the condition of hypotonicity. This is detected by the brain and this produces a signal which is known as now the control center is our brain, which integrates the data and produces a hormone known as ADA, produces less of the hormone known as ADH, which is antidiuretic hormone. This hormone essentially carries the signal from the brain to the kidneys. This signal, what it does is that it increases the amount of it decreases the amount of water reabsorbed by the kidneys, and this since there are low amount of water being reabsorbed by the kidneys back to the blood, a lot of volume of water is being excreted out, thus decreasing the overall water content of our blood, and this causes water content of the blood to go back to normal. In the opposite condition, in which you have been eating a lot or sweating a lot, water content of our blood decreases. This allows the body, this information when it reaches the brain produces more ADH or antidiuretic hormone, which causes the kidney to reabsorb a lot of water from our digestive tract and the blood, and this causes a low amount of urine being produced, and because all of the water has been taken out of the urine and placed back into the blood, so our blood, the amount of water in the blood rises again to its normal condition. This is how the kidney works in order to maintain the optimum salt and water balance in our body. Any questions for this? So this is a bit complicated. Take a bit of time and go through it and think if you want to ask anything from this. Okay, Ria Kolekar is asking. She has joined late and wants to ask what topics have been covered. So we have, Ria, we have been talking about a lot about the assignment questions that were being. We are actually going through the assignment questions of previous years and trying to answer various questions from this and trying to understand the concept that we are dealing with behind the questions. So we have already covered a lot about homeostasis, feedback mechanisms, as well as homeostasis, feedback mechanism, temperature regulation is some the parts we have covered mostly for till now. We are now going about a different kind of feedback mechanism we are discussing now is osmoregulation. If you have any questions regarding the previous one, you can put them into the chat, we can discuss them. Okay, anyone has any questions from this? If not, then we can move forward.
No, sir.
Okay, so the answer to this question, dash is the process of maintaining salt and water balance across membranes within the body. The answer is osmoregulation. Okay, so next we are diving a bit more deeply into the salt water balance in our body and asking a question, which is optimum sodium ion concentration for extracellular fluid will be how much? The options are 138 to 146 millimolar per liter, 230 to 500 micromolar per liter, 230 to 500 millimolar per liter, or 138 to 146 micromole per liter. If anyone has any idea about this, they can put the answers right directly into the chat. In access, it is C, 230 to 500 millimolar per liter. Vashishta says it is D, 138 to 146 micromolar per liter. Muskan is also D. Jisu says it is A, 138 to 146 millimolar per liter. Ramanathan says it is A. Vashishta says it is A. Aruna is also going for A. Anyone else? Anyone else wants to try and answer this? Optimum sodium concentration for extracellular fluid. The RIA also says it is A, 138 to 146 millimolar per liter. Okay, so we'll go forward and go through this. So these are the, so when we were previously discussing about the solute concentration and the tonicity of an osmoregulation, we had been talking about an overall salt concentration. We were not discussing the specific ionic concentration of different ions, which is something that we are going to discuss now. So as you can see, as compared from the intracellular to the extracellular fluid, different ions such as potassium, sodium, chloride are actually present in different concentrations. So potassium is present much higher concentration inside the cell as compared to the outside. The opposite is true for sodium. Much lower concentration inside the cell as compared to outside. Similar trend is followed by chloride. Much lower concentration of chloride inside the cell as compared to outside. This is a way, so for every one of these ions, the same principle applies. If there is a disparity in concentration, always the ions will try to move from one side of the cell to the other side. So all the potassium ions are always trying to get out of the cell, whereas all the sodium ions are trying to get inside the cell. But this disparity is actively maintained by the cell because of various reasons. The major one being to maintain its salt concentration, as well as something known in case, especially in case of nerve cells, in order to maintain their surface potential. So as we know, all the cells in our body, they have a particular charge. Most of cells are negatively charged. This charge is due to the concentration differences between the salts that are present on both sides. Also, this concentration gradient is used to transport various other molecules across the cell surface. This is a bit advanced concept in which, say, a particular solute, say glucose, has to come from outside of the cell to inside of the cell. Cells always have a higher concentration of glucose inside, so glucose does not want to come in. But what happens is that there is a coupling of sodium entry and glucose entry into the cell. So sodium, which also always go into the cell, helps transport of glucose inside the cell. This is something that is very important and is known as cellular transport. All of this is maintained via this actively maintained disparity between concentration of ions. And this is done by something known as ion channels. So the entire cell membrane is made out of something known as lipids, which are water insoluble molecules. These but in between this, there are channels or pathways for ions to go across. These are known as ion channels, and these can be specific for some particular ion to go in or some particular ion to go out, and these can be controlled. And using these channels, the cell controls what exactly would be the concentration of ions outside the cell and what will be the concentration of ions inside the cell. So this was a lot of information, but essentially the takeaway from here is that there is an actively maintained disparity between ions inside the cell as compared to outside of the cell, and this is very important for cellular transport and is maintained via various ion channels present on the cell membrane. Okay, anyone wants to ask anything from this? So I will give you a minute to go through this because it is a complicated part.
Let's move to the next one.
We can move to the next question. Yeah, so this is clear. I will still give everyone else a moment to go through this because this is a bit complicated. So we'll wait a minute and let's see if anyone has any questions.
Okay, sir.
Hi, do we have to study like all of the ion concentrations of like given potassium, chlorine, everything for the exams?
Not really. This type of question being asked is bit uncommon, but I would really recommend having an idea about potassium, sodium, and chlorine because these are the major three, and these always have a similar-ish values on either side. So potassium and sodium is just the opposite of each other, 140 to 150 outside or inside, as compared to 4 to 5 to 10 to 40. Chlorine is also almost similar. So these are the major three that we discuss most of the time: sodium, potassium, and chlorine. Knowing these three would be enough. But essentially from this slide, I would like you to take away the concept that we are discussing here about the actively maintained disparity between the ion concentrations and the concept of ion channels. Okay, so since we do not have much questions from this, we'll just go ahead and answer the question. The optimum sodium concentration for extracellular fluid is 138 to 146 millimolar per liter, right, following the previous slide. Okay. Any questions from this? Because we will be moving forward a bit and moving away from this, and we'll be talking about the molecular level of organization that we had that was discussed during the lecture notes in the normal lectures, and how exactly. Yes, anyone has any question?
Sir, in exam, they will not give the question from too much deeply, like deep concept.
I think most of the questions will be conceptual. So you will be asked to your thinking capacity will rather be tested than your knowledge of these values. But this you will see most of this in upcoming assignments, how exactly we have the questions are framed, and even the first week's assignment that is for this year, if you go through it, you will see that most of it is based on how you think rather than what you know. That is the major idea here.
Yes, sir.
Okay. Okay, so if we are done with that, we will go on to the next question. The phosphate end of the phospholipids. So we have now moved from homeostasis, feedback mechanisms, osmoregulation to another part, which is the molecular level of organization in physiology. In this particular question, we will be having an in-depth discussion about various molecules of life. And first, I would like to go through the question and ask everyone to try and answer this. The phosphate end of phospholipids is dash and fatty acid end is dash. The options are hydrophobic, hydrophobic, hydrophilic, hydrophilic, hydrophobic, hydrophilic, and hydrophilic, hydrophobic. So if anyone has an idea about this, they can go ahead and answer. Otherwise, we will discuss it. So Bisu says it is C, hydrophobic, hydrophilic. Sharan says it is D. Vashishta is also saying it is D. Ramanathan also says it's D. Anyone else? Devi also says it is D. Ria is also saying it is D. Sanjana and Ashna also say it is D. B, so changes his answer to D. Nice. Okay. Anyone else wants to try and answer this?
Sir, A option because uh, the phosphate is having a hydrophobic tail and the fatty acid is hydrophilic in nature.
Let us discuss. We will go through the entirety of it. So, uh, okay. So, one of the final lectures called for everyone to go through the various biomolecules that make up the molecular assembly of cells, and I, for betterment of everyone else, I have compiled various information about the four types of biomolecules present. So we will have a long discussion about the various biomolecules. If you have not come across this will be very helpful to study in other courses as well. So keep an eye out for this. And if you have any questions, because you have not gone through this beforehand and want to ask, you can stop me and ask anytime. So there are four molecules of life which are known as biomolecules, which the cells use for various types of its to achieve various functions. These include the first type, carbohydrates, which store short-term energy and are used as building materials; lipids, which store long-term energy and used in membrane formation; nucleic acids, which store hereditary information for constructing and assembling of various enzymes; and proteins, which are the workhorses of the cell and do essentially everything else that the cells wants to do, which involves metabolism, various enzymatic reactions, even movement of the cytoskeleton is done by the proteins. Hello, am I audible? Okay, hello, am I audible? Am I back again?
Yes.
Okay.
Sir, now you are too much clear than the that one.
Mm, we have a lot of, wait, I will start sharing my screen, then it should go. Yeah, is the PPT visible? The PowerPoint is visible, right? Hello,
Yes, sir.
the PowerPoint is visible, right?
Yeah, yeah.
So we were discussing the four types of biomolecules. The carbohydrates, which store short-term energy and are used as building materials; lipids, which store long-term energy and are used in membrane formation; nucleic acids, which are store hereditary information for constructing and assembling various enzymes; and proteins, which actually do everything else that the cell requires it to do. Proteins are essentially the workhorses of the cell, which control from metabolism to cytoskeleton rearrangement to movement of the cell to everything else that the other biomolecules cannot. So we'll be going into depth about all of these biomolecules. All the information is present in the PPT. I will be glancing over this information for now. You are free to look at it at your leisure after the lecture, and if you have any questions about it, you can come back to me in the next class or in the NPTEL forum. So the first class of biomolecules are known as carbohydrates. As the name suggests, these are molecules present made out of carbon, and hydrates essentially points to the general formula of carbohydrates, which is CH2O, that is a carbon and water. But if you look at the structure, they essentially have a long chain of carbon molecules and various organic groups, which are essentially all alcohols other than one major aldehyde or a ketone group. So the most common type of carbohydrate is known as glucose, which has an aldehyde group at its top and has a six-membered carbon chain. Here the orientation of the OHs is important because this actually represents the three-dimensional orientation of the molecule, and a OH on the right side essentially indicates that the hydroxyl group is inside the plane of paper, whereas OH on the left side says it is coming out of the plane of the paper in 3D. This representation is known as Fischer representation. But all of this is not very important. Essentially what we want to know from here is that carbohydrates are made out of long carbon chains containing hydroxyl groups and aldehyde or ketone groups. They are used majorly for energy production and are used in metabolism. So we have the structure of carbohydrates. So these are the simplest carbohydrates present, which are known as monosaccharides, which only have one chain of carbon attached via covalent bonds, and these include glucose, fructose, galactose, all simple carbohydrates, and these can be directly used for production of energy. Since they have a lot of hydroxyl group, these are polar molecules which are easily soluble in water, and that is why you see sugar dissolving in water very easily, because it is made out of glucose. For now, this much information should be enough, but for your information, there are other classes of carbohydrates which are much more complex, which are known as disaccharides or polysaccharides, which are made out of multiple of these chains and have various functions from metabolism, energy production to production of support structure of the cells, which help in its rigidity. If anyone has any questions from carbohydrates. Okay. So moving forward, we have the second class, which are lipids. Lipids are essentially one hand carbohydrates are soluble. Lipids are non-polar biological molecules which are not soluble in water, but can easily traverse the cell membrane because of their non-polarity. There are various classes of lipids. The first class being steroids. Steroids look something like this. They have a uh, four cyclic chains attached, four cyclic groups attached to each other, which look like this. The simplest is cholesterol, and adding various modification to cholesterol, we have different hormones such as testosterone or estrogen, and used in as these are used either in metabolism or as hormones which produce signal, which transfer signals from one part of the body to another. The other major class of lipids is known as fats and fatty acids. These are a bit complicated. These have two parts. First is known as the glycerol molecule, which is this orange part shown here. This has a CH2 and triacyl normally glycerol essentially has a hydrogen here. So it is a CH2OH, CHOH, CH2OH. So it has three hydroxyl groups attached to a single carbon backbone. In case of fatty acids, we essentially have removed that H and form a acyl chain. Acyl chains have long chains of carbon molecules which have a acid and at one end, which forms a ester bond between the glycerol molecule and the fatty acid chain. So for example, we have stearic acid here, long chain of carbons, and one end we have a acid. This acid forms a ester bond with the OH of the glycerol molecule, essentially forming three hands of the fatty acid or the fat, where three hands are non-polar and hydrophobic, and other three hands are attached to the glycerol molecule. One important type of fat is known as phospholipids, in which the one of the three hands of the triacyl glycerol is attached to a phosphate group, and there is a small polar group attached to the phosphate group. So it looks something like this. But the other two hands have long carbon chains of fatty acids attached to it. So this is known as a phospholipid. Here we have the phospholipid phosphatidylcholine. The name is not important, you just have to understand that what exactly the phospholipid is made out of. It has two parts. One part is the hydrophobic end, which has long chains of carbon atoms, and the other part which has a hydrophilic group, which is ionic or polar and which loves water. So one part loves water, the other part hates water. This kind of phospholipids are the major help in the major construction of membranes. In case of membranes, the hydrophobic parts are hidden inside and the hydrophilic parts are exposed to the intracellular and extracellular aqueous surfaces. So essentially phospholipids form membranes and have two parts, one hydrophilic and one hydrophobic. Third, we have the proteins. The proteins are essentially macromolecules that do everything in our body, from carrying out enzymatic actions, gene activators, contractile filaments, molecular motors. Everything is done by proteins. For example, these are feathers of a bird. These are also made out of proteins. In case of eyesight, this is also controlled by proteins. So proteins are made out of small molecules known as amino acids. Amino acids have a general structure which look like this. They have a central carbon atom known as the alpha carbon joined to an amino group on one side and a carboxyl group on another side, and having a side chain. Depending on the side chain, the amino acid can be polar, non-polar, polar or non-polar, and can be classified in different parts. Various these amino acids can join together and have a reaction among themselves to form a peptide bond to form long chains of amino acids. These long chains of amino acids actually form the proteins. So long chains of amino acids form the primary structure. Subsequent work is done on the protein in order to form the secondary structure, the tertiary structure, the quaternary structure, or supramolecular assemblies. The quaternary and the supramolecular assemblies of the proteins are actually what are the functional final forms of the protein that actually do everything else. And quickly moving on to the nucleic acids. A nucleic acid contains, or we know what nucleic acids are probably. These are two types, DNA and RNA. They have various DNA and RNA are made out of several small nucleic acid monomers, which can be which are four types: A, T, G, C, adenine, thymine, guanine, or cytosine, and this essentially code for the entire genetic sequence or the genomic sequence present in our nucleus of the cell. Yeah. Here we have jotted down some functions of nucleic acids. So a lot of information in a very quick time, because I do not have this, because this is a pretty complicated part and has a lot of information. I would rather have you go back and go through the slides and see if you understand everything. You do not have to understand all of this. Basic concepts of how exactly all of this function is what you require, like what they look like and what are their functions, is all we need from this. Any questions?
No, sir.
Okay, so coming to the initial question that was asked, the phosphate end of phospholipid is dash and fatty acids is dash. And if we go through the material that we just discussed, then we'll know that the phosphate end of the fatty acids is hydrophilic or loves water, and the fatty acid end is hydrophobic or hates water. Is this fine? This is
Yes,
a bit complicated because we have a lot of information being bombarded at that time. So I will really like you to go through the slides once we are done here to better understand this.
yes, sir.
Okay. Next, we have the next question, which asks, glucose is a what kind of biomolecule? Protein, lipid, nucleic acid, or carbohydrate? So you guys can put in your answers here. Aruna says D, Sanjana D, Vashishta also says D, Sharon also says D, Akansha also says D, Devi is also with D, Maheshwari also says D. Anyone else wants to answer this?
Carbohydrate.
Yes, you are correct, Vinayak. Glucose is a carbohydrate. Ashna also goes with D. Okay, so we have just gone through this. Carbohydrates are one of the biomolecules, and glucose, as you can see the structure right here, is a carbohydrate which has a aldehyde group on its top and 6 carbon long chain, and it is the most abundant carbon monosaccharide present in the world. So glucose is a carbohydrate. Next, in the next question we have, how many carbon atoms are present in one molecule of ATP or adenosine triphosphate? The options are 1, 5, 10 and 50. If you have any idea, then you can just put into the chat and say what you think is the correct answer. Do we have answers coming in? Aruna says it is D15. Akansha says it is C. Vashishta says it is C10. Sharon says it is D15. Anyone else wants to try and answer this? Aishu says it is D15. Okay, so we'll go ahead and discuss about ATP. So ATP is a nucleic acid known as adenosine triphosphate. So nucleic acids are, as if when you will go through the entire part on nucleic acids, you will know that nucleic acids consist of three major parts. One is the nitrogenous base, which can be ATGC or AUGC, adenosine, adenine, guanine, thymine, cytosine, or uracil, which determines what kind of nucleic acid it is. A ribose sugar backbone, as you can see, ribose sugar base, as you can see right here, which is essentially a 5-membered ring, which is essentially a carbohydrate, and a third part which consists of phosphate ions joined to each other. So in case of nucleic acids or RNA or DNA, there are various parts or various of these molecules joined to each other via these phosphate groups. You have adenosine monophosphate, guanine monophosphate, thymine monophosphate, or ATGC, everything in one line. But when they are alone, they actually serve in another purpose. Adenosine triphosphate serves to be the energy currency of the cell. ATP can produce energy at will, and the energy is utilized by the cell to perform its various functions. Most of the time, ATP is every one of the metabolic processes tries to produce ATP, and then that ATP is used by the cell to perform its various functions. So this entire molecule is ATP, consists of the adenine, the ribose, and the phosphate. And the ATP, the T is triphosphate. So when there are three phosphate molecules attached, then it is triphosphate. Two, then it is diphosphate, and one, it is monophosphate. The red colored bonds here shown are very high energy bonds. In order to release energy out of ATP, these phosphate bonds are broken, and that produces a lot of energy, around minus 31 kilojoule per mole of energy, that is then utilized. So this is ATP. This we will be encountering it when we will be discussing about a bit about metabolism going forward, but essentially this is it. And to count the number of carbon atoms, as the question was, we can just count it here. It is 1 2 3 4 5 6 7 8 9 10. There are 10 molecules of 10 molecules of carbon present in ATP. So the answer is 10. Any questions from this? Okay. So that was all for today. We have covered the first week of assignment questions from the previous year, and we have covered various topics including homeostasis, feedback regulation, osmoregulation, then talked a bit about biomolecules of life, and Aruna is asking what pattern will the exam question be. It will most probably be a MCQ, but you can again ask it in the NPTEL forum, because it will have the TAs from the course as well as the professor there, and they will be better able to answer what the final exam pattern will be like. But the assignment questions will be MCQ, and most probably the final exam will also be MCQ. But it will be an offline exam. We will have to travel to a exam center, is what was previously also the case. You will have to travel to an offline exam center. You will be able to select which center you want to appear in via the exam form that you fill at the NPTEL site. Okay. If okay, so if no one has any other questions, then we will be ending the. Yes, I will take it in. Just wait a moment.
Does the attendance matter?
The attendance actually for these actually do not matter. but you are encouraged to actually come to these lectures because this will help you a lot to understand how exactly to answer all the questions in the final exam as well as the other this year's assignment questions. And these live sessions were actually designed in a way in order to help you out, so that you have any questions that you cannot find a very good answer in the text or the forums, then you can ask them directly in these kind of sessions. So being prepared, going through the previous week's lectures and asking questions is how you can best benefit from these classes. I joined today onwards, so I couldn't do last week's assignment. So you will have, so the last week's assignment, the final date for submission of it is actually in the coming month. It is probably 7th of the coming month. So you still have a week to do the assignment, and I definitely recommend doing the assignment because it counts towards a huge part of your final grade and will help you out in passing the exam. If you do not have enough marks, then you will not be able to secure a final exam certificate. So you still have time to finish it. You please make out the time and do it. Okay, if anyone does not have any more questions, then we will end the session right here.
Yes, sir.
I have a question. One second.
Yes, sure.
Yeah, I'm currently pursuing my bachelor's, so I'm in my final year. So I'll be having exams in the set date about 26th of October. It is written on
Okay.
the side. They're having exams. So there's any possibility we can change the exam date or something for?
Changing of exam date is very difficult because NPTEL conducts one exam at the same time at the same date throughout India. But you can actually put in a mail if you want to the to NPTEL via the forum. If there is any possibility of an alternate exam date, if they are having exams on two different dates, then you can maybe attend at a different time.
Okay, fine.
Now, if we do not have anything more, then I will be closing the session. Thank you everyone for joining. See you next week.
Yes, sir.